There we go. Okay. And we had talked about polynucleotides as the second kind of category of polymer that we're going to be discussing in greater detail in living systems. And we mentioned all of this last time, so I'm going to go ahead to the next one. Polysaccharides are an interesting contrast to both proteins and polynucleotides. In general, when you find polysaccharides, they will be homopolymers, the same sugar moiety throughout, although some of the sort of oligosaccharides on cells are pretty heterogeneous. Polysaccharides have a number of functions that we tend to not give enough respect to. Obviously, there's fuel storage. You have in your body a certain amount of glycogen, and that glycogen is what allows you to keep going on a regular basis. Little kids have only a little bit of glycogen. That's why you have to keep giving them juice if you want them to walk very far. Polysaccharides are also preeminently good for structural support, and we see that especially in the prokaryotes and in the plants. Our structural support tends to be in the bones, but trees depend very heavily on it. Polysaccharides are mostly linear. I guess if we're going to talk about cellulose being the most abundant one, I would say almost always linear, but we'll see where the branching is useful. Polysaccharides can carry a certain amount of information, and so they're not exclusively the dumb slave of the proteins, the nucleic acids. But they are not as diverse in their structural forms as polypeptides. If you think about what they have to work with, we're talking about basically polyalcohols. You just can't get the kind of variety of chemistry that's possible for the polypeptides. This is showing you one particular format for glucose, and you can see the alcohol group sticking away from the plane of the sugar. And we'll talk a little more about that structural chemistry when we get there. This is an example of a starch molecule, and this is a cellulose. It's necessary to think at least a little bit about what's sometimes called physical chemistry in order to understand where living systems get the energy to live. So one of the important things we're going to be talking about when we talk about metabolism is energy, where it comes from, what it's used for. And in all cases, a reaction has to be driven in some way by free energy. So what does that mean? Well, Gibbs was a brilliant theoretician in the 19th century who had wonderful insights into physical chemistry and energy and so forth. And he was the one who has been recognized in this formula for free energy, that Gibbs free energy is the difference between the enthalpy, which is the heat content, how much the molecules in there are bouncing around, minus the entropy, the disorder, times the temperature. So this is a way of describing the energy content of a system. It turns out to be remarkably difficult to do the energy content quantitatively for a system because of trying to keep track of it. What tends to be easier is looking at the difference in free energy, starting someplace and going somewhere else, and talking about the difference in free energy between those two states. So if we look at this A here as one state, and here's another state, someone has obviously hit that with the cue ball, you can see that this has more disorder. And you know that if you were to do this 17 times, it would be a different pattern each time, and that's part of what is the meaning of its having entropy, having disorder. And so it is spontaneous for a reaction. Come back a little. It's spontaneous for a reaction to go from here to there. There's a loss of free energy. It would not be spontaneous to go from here back. If you saw a video of it going from this to that, you would know either the movie was running backwards or that somebody was fiddling somehow. Give me a second while I get the pointer on permanently. There we go. And so whenever the products have less free energy than the starting thing, that can be considered a spontaneous reaction. It won't say anything about how fast it goes. It simply says there's enough energy for it to happen without other energy being put in. Now, there are going to be reactions we want to have happen that don't have a negative free energy. Fortunately, because of thermodynamics, you can couple reactions so that you can drive one with the other. So if we have a reaction like A goes to B, and it is a spontaneous reaction with a negative free energy change, and there's another reaction from B to C, which has a positive free energy change, we can get from A to C because overall, the sum of those reactions is a -5. It has a negative free energy change. This is possible because B is the common intermediate. Obviously, if it was A to B and D to F, you can't add those reactions together in any meaningful way. So this always depends on there being a coupled physical character to the two reactions. And this energy is used for basically oxidation and reduction reactions. So if you look at carbon dioxide in the air, here you have the carbon dioxide, 2 double bonds to each of those oxygens. This is a very oxidized carbon. That's as oxidized as a carbon can get. And going from that to a reduced state is not a spontaneous reaction. It's not a favorable reaction. Somewhere or other, there's got to be some energy put into the system to make it go in that unfavored way. And in the real world, almost all that energy is going to come from light energy and through photosynthesis. A few little things live down in the steam vents off of sulfur, but mostly it's from light. And here we have a carbon that's been partially reduced. Now that it's reduced, it has it can provide free energy to another reaction. So as it goes back to an oxidized state, that potentially leaves free energy available to do something else. This is just a reminder for you of the oxidation states of carbon. I know this is a very small print on the screen. I don't expect you to read it off the screen. But I'll give you sort of the highlights of it. At the very top of this scale is the carbon with 2 oxygens, the most oxidized state of carbon. And then down at the bottom, you have a methane molecule, carbon with 4 hydrogens. That's the most reduced state of carbon. And you can go through in between and get the general sense of being more or less reduced as a carbon atom. And if you remember the core relationships between carbon, hydrogen, and oxygen in electronegativity, you can use that as a way to remind yourself of which one is which. Any questions about this before we go on? We call that state the most reduced state because it's the one in which carbon is getting the biggest share of the electrons. So its overall energy state is more negative than it would be up here. Up here, because oxygen is more electronegative, it's getting the lion's share of the electrons. It's not an all or nothing thing, it's a shared thing because these are covalent bonds, but there's winners and losers in the reaction molecule. So where do these systems come from? Some very interesting experiments were started a while ago trying to get at the idea that maybe some of these molecules could occur spontaneously. And you may have seen these reactions where you start with a mixture of gases and put energy in the form of artificial lightning through the electrodes, let that run for a while, and then take whatever's out, come out. It was an interesting experiment. It definitely produced carbon compounds that were more complex than the inputs. But there were some difficulties with it, and one big difficulty was the early atmosphere of the Earth was probably not as reducing an atmosphere as the original experiments used. But there was a variety of molecules formed, and even under other conditions, a considerable variety can form. And we know that this is something that not merely can happen, but does happen, because in space we have observed quite a large variety of these compounds, either on meteorites or spectrally in the systems of planets and stars. So it is possible for a certain level of complexity just to arise spontaneously. The more recent thoughts about where life might have started and what it took for life to get going have focused often now on hydrothermal vents. Hydrothermal vents are these things in the deep ocean where underneath the crust, volcanic areas are causing the stone to heat up, and then the water above it gets very hot and so on. And the advantage of there are several advantages to hydrothermal vents. One is that the heat that is produced through the volcanic processes provides a free energy change, so that's a free source of energy. In many of these hydrothermal vents, there are reduced compounds around, so you get away from the problem of not having a reducing atmosphere. The vents are always in close location. They're nearby to places that are cooler, so you have a place where you could have things stabilized in a cooler environment, or you could take advantage of the thermal gradient. And indeed, we have found in the areas of these things like acetic acid and short polypeptides that are forming abiotically without biological interference. Some of the other proposals that have gone on about how life might have started have invoked clay-like surfaces that to get things organized, it would be nice to have somewhere to get it started. And one of the proposals is that clays, which have an array of positive charges on them from their chemistry, spontaneously formed geology type stuff, that could provide a location at which negatively charged monomers could sit down close to each other, and because they're close to each other, be more likely to become a polymer that then can leave the clay and go off and try and do something. And there are a number of people who are looking at just what kinds of chemistry, in fact, are possible from clays. It does seem that the organisms that live in these vents are, many of them, life forms that have persisted for a very long time. So this is a place where things 1 billion years old go on making a good living. Sorry about that. In order for something to be alive, there has to be, either at the beginning or the end of it, a process that you can call replication. That replication is the essence of life. You have to be able to copy information imperfectly so that you can have selection from then on. But you need to have replication to have a system that can be called alive. And this is pointing out the idea that some people have favored an early form of life that might have been based on RNA rather than on DNA, where the genetics are on the RNA. And this is just a schematic of what that might look like. If you had a polymer of A's, you'd have, through just the hydrogen bonding used matching up, those chains could each now make a complement to their sequence, and you'd end up then with more than you had to begin with. That diagram is called a mechanism in the figure legend. It's not a mechanism, it's just describing what it means to replicate RNA. But there is very interesting evidence in recent years, and one of the things that has been discovered are called midwife molecules that can slip in between the bases and act as non-biological catalysts for polymerization. So these are molecules that are found in the environment that can facilitate the polymerization of RNA from monomers. Very, very interesting work, and I will try to post on the website articles if people want to read more about that. The other difficulty I had always felt with the idea of RNA life as a starting form is that RNA is not terribly stable. I don't know if any of you have worked with RNA yet, but it's kind of like, your crazy cousin that you try to be real gentle with because they're going to break up otherwise. And it turns out that in the environment, there are minerals that stabilize RNA dramatically. And these are the borates. You've probably run into the borates in the context of borax. People use that to wash their clothes. And the reason it washes clothes is because it complexes with carbohydrates. And so the borates can stabilize the RNA molecules and prevent them from turning into tar. So it opens up the window of what might be possible with an RNA chemistry. Any questions about that before I move on? Okay. So we've talked a little bit about what kind of things might have gone on in the early stages. For the first cells to exist, somehow they had to figure out a way to fix carbon. What we mean by fixing carbon is taking inorganic carbon in the environment and modifying it chemically so now it's part of an organic molecule. So you can increase your mass from what you were before. So someone somewhere had to invent a way to fix carbon. We don't know specifically what the first organisms were to do that, but we do know the organisms that did a lot of it. The blue-green algae, which are also called cyanobacteria, invented photosynthesis a very long time ago. They were actively making large structures in tidal flats called stromatolites about 3.5 billion years ago. And 3.5 billion years ago, they were fixing carbon and they were doing photosynthesis that produced oxygen. So they were among the earliest things to be alive and to modify the environment. They were probably not the first, but they were among the very early ones. Broadly speaking, we can divide the prokaryotes into two groups. We can talk about the eubacteria and the archaebacteria. The archaebacteria is a group that was given that name because the man who described them as a group thought they were among the first bacteria. They include things like hyperthermophiles and methanogens and things like that. May not, in fact, be any older than the eubacteria. If people are interested in that, I can give you references on that. But they are distinct from the other broad division within the prokaryotes. You may notice a certain inconsistency about K's and C's in the way I spell prokaryotes and eukaryotes. I think I got the English spelling fixed in my head and I can't always correct it. As a general principle, if I in the book disagree, if I haven't specified that I'm differing, you can wave your hand and say, but the book says. But in general, if I say something that's different from the book, it's because I think I'm right and I think the book is wrong. And you're quite welcome to bring up references that show that I'm wrong. That's cool. That's not a problem. But if there's a time when I've said something in lecture that's different from what's in the book, what I've said in lecture has priority over what's in the book. And so I wanted to give you a more developed, a little more subtle understanding of the contrasts and similarities of prokaryotes and eukaryotes. Because what's given in your textbook is kind of the standard 3-sentence contrast, which if you're talking to, a 14-year-old would probably be enough, but you're not 14-year-olds. You're juniors and seniors, and you can handle the truth. Prokaryotes, as a great group of organisms in general, have single or no internal membranes. The book says no internal membranes, but in fact, many prokaryotes have internal membranes of one kind or another. Blue-green algae have internal membranes that are involved in photosynthesis. Planktomycetes have one involved with this peculiar way of using ammonia for a metabolism or energy source. But they're single. You don't see the kind of double-layered membranes that you see around chloroplasts, mitochondria, or the nucleus. So single, maybe a lot, but always a single membrane. I think the idea that they don't have internal membranes came from the fact that most of the ones you hear referenced are basically parasites. E. coli lives in our gut. It has a very boring life. It doesn't need to do anything terribly complicated. And so I think that the interesting, complex, confusing prokaryotes like blue-green algae and planktomycetes, those are free-living and they've got to do things to make a life for themselves, and they have the membranes and stuff. Many prokaryotes have some kind of a wall, a peptidoglycan of some kind. The genetic information for prokaryotes is in a circular DNA, complete, continuous, covalent circle. And it's packed up quite a bit in an area called the nucleoid. So it's condensed down to fit in there. Many of these organisms have a DNA circle that's as large across as the cell, so obviously they've got to pack it quite a bit to fit it in. The cells themselves are usually small, like less than a micron, but not all of them, and we'll talk a little bit about that. They have very complex biochemistry. You can find prokaryotes that will metabolize just about anything. You can find prokaryotes that, poop out gold. You can find prokaryotes that will metabolize testosterone. They did that by isolating soil samples from fire hydrants. I didn't make that one up. There are prokaryotes that will metabolize uranium. So they're really extraordinary. And prokaryotes are the organisms that are at the extremes of life. They can live in boiling water. They can live in pH 2 solutions. They can live in the soil in the water of the Salton Sea. So they have the capacity to adapt biochemically, metabolically, to extraordinarily extreme circumstances. They can live below freezing. Very strange. They can be multicellular. They're not all unicells. Not only that, some of them have differentiation, meaning they have more than one cell type. Not only that, some of them have terminally differentiated cells, cells that have taken on a function and will never divide again. So prokaryotes, some prokaryotes, have in fact a considerable spectrum of behavior open to them. Now, in the ones that are multicellular or differentiated, there are never very many cell types. I think the highest count I've come up with so far is 3 cell types. There may be more. If there are microbiology majors that want to correct me, please do. But they do differentiate. It's not quite as simple as people tend to make it. Any question about these guys before we go on? Okay. I like this picture because it gives you a sense of scale. And for all of these things, there has to be some caveats, and we'll add those in later. But this is an example of virus particles. You can see a scale bar here of 300 nanometers. This scale bar is 175, so it's this is about half the length of that. You can see that the prokaryotes here, which I think are E. coli, are way bigger than these viruses. And then you have almost the same scale bar on these two, and you can see how much bigger the red blood cell is than the prokaryotes. So it gives you an intuitive feeling for the general scale of these things. I don't want to mess with your minds too much, but there are viruses that are as big as these cells. And we're going to see prokaryotes that are way bigger than this. But this is a good generic scale sense for you. Eukaryotes. All eukaryotes have internal membranes. They all have internal membranes. And they have those around diverse organelles, quite a remarkable diversity of organelles. A few of these organelles have double membranes, the mitochondria and the chloroplast, and the nucleus. And the mitochondria and the chloroplast also have DNA. They have genes of their own. The nucleus does not just have a double membrane, it has pores in it that are unique to the nucleus that allow RNAs to go out and proteins to come in, and for that matter, ATP to come in. There is no protein synthesis in the nucleus itself. Now, I make that point as a strong point because that is, so far, an absolute contrast I've been able to find between prokaryotes and eukaryotes. That in eukaryotes, there is no protein synthesis in the compartment of the genome, and in prokaryotes, there is always some, sometimes all of the translation goes on in the same compartment. Another contrast is eukaryotes have linear chromosomes. You remember I talked about a circular chromosome for the prokaryotes. Eukaryotes almost always have linear chromosomes. Some big change in organization there. We're going to see that linear chromosomes present a specific challenge in how they cope with it. Eukaryotes may or may not have a wall. It's fairly variable. If they have a wall, it's usually cellulose rather than the peptidoglycans of the prokaryotes. In this figure here, I've taken the E. coli, and here is the paramecium. Those are at the same scale, all right? This thing may well eat some bacteria, and you can see how it's big enough to do that. So this also may give you a little bit of a sense of scale. There's a very wide range of sizes to eukaryotic cells, much wider range than there is for prokaryotes. They have more complex morphology than prokaryotes. And because of those things, almost certainly, they are not able to stand up to the nasty conditions that prokaryotes can. That the limits of temperature, the limits of pH, and so forth, for the eukaryotes are narrower. So the prokaryotes can use metabolism to be tough guys in extremes. Eukaryotes use complexity of structure to accomplish things you can't do if you're too small. I wanted to give you a sense that it's not all or nothing, and this is my attempt to sort of convey some of that. This is, if you notice, at a logarithmic scale. So here's 1 micron, here's 10 microns, here's 100 microns, 1,000 microns, or a millimeter. There's a meter over there, okay? And so, although it's true that the great majority of prokaryotic cells are down in this range, a bit less than a micron to a few microns, There are definitely quite a number of cells, prokaryotic cells, that extend up into the larger ranges. And I may talk about a couple of cells that actually end up almost a millimeter across as a prokaryotic cell. So these upper end prokaryotes here are bigger than most eukaryotic cells. So size is not an absolute distinction. Eukaryotic cells have, I think, really a greater variety of sizes. Some of the eukaryotic cells are almost as small as prokaryotes. In the last 10 years or so, we've discovered that most eukaryotes are smaller than 3 microns. When people in the oceans would do oceanography and use a net to capture the cells, they use like a 10 micron mesh and they come up with some algae. And then they started using a 5 micron mesh, and then there was other things that they've been missing all that time. And then they started using a 2 micron mesh. And it turns out that there are eukaryotic cells that are smaller than a micron that comprise up to a quarter of the biomass in some places. So the atmosphere that we breathe is in part due to organisms that small. So, on the other hand, they get quite big. In this range, we're talking about things like oocytes and frog eggs and fish eggs. It can be 10 millimeters across. And you might be wondering why I'm showing eukaryotic cells as long as a meter or 10 meters. But neurons have to get where they're going. And if you think about how big a blue whale is, the neurons in a blue whale are very long. If we look at the size range of multicelled eukaryotes, we can see that the smallest of them is actually smaller than the biggest prokaryotes. So you can have things like rotifers or some of the smallest insects that are actually smaller than these big prokaryotes. Multicellular eukaryotes that are smaller than the biggest prokaryotes. I have it over here going up into, if you look carefully, that's kilometer ranges. And the organisms that can go up to kilometers are things like fungi and aspens that make a clonal colony that's genetically identical that can be a kilometer or more across. Mammals as a group are more narrow in their distribution, and here we can see an absolute difference. The smallest mammal is bigger than the biggest prokaryote. So we finally have a place where we can make a thing. And you can see that the range of the black size is also smaller for the mammals. So this is maybe more information than you wanted, but I think that the textbooks tend to make it sound very simple. And I think the fact that we are discovering, in the last decade, organisms that may comprise 10% of the biosphere is a sign that we don't necessarily know everything that's going on. This is giving you some names for those various ones. I won't walk you through all the details, and I certainly don't expect you to memorize them. This is just for people who want to know what was on that thing. But this little bitty thing here, the picoalgae, Ostriococcus, is this eukaryote that's less than a micron across. It's about 0.9 microns. And it has 1 chromosome, and it has 1 chloroplast, and 1 mitochondrion, and 1 stack of Golgi. So it's just got 1 of everything. Prochlorococcus, which is the most abundant blue-green algae that was missed until a decade ago, is only 0.6 microns. So they're just that different in their diameters, but if you do the geometry, the eukaryote is 3 times as big as the prokaryote. So the smallest prokaryote is smaller than the smallest eukaryote. And this is giving you some pictures of those guys. And here you can see a blue-green algae. This is a prokaryote. This is also called cyanobacteria. This has internal membranes. It has lots of them. You can see them all over the place. You can see these specialized little thingies that are inside of it. What it doesn't have is a nucleus. It doesn't have that double membrane-bound nucleus. And it doesn't have a chloroplast that's a discrete compartment. This is a picture of Ostriococcus, that smallest one, where you can see it's 1 nucleus, 1 chloroplast, and so on. So this and this is actually quite possibly smaller than this. Cells are not the cells we see today are not the cells that have always been there. They evolve. And I'm going to talk a little bit about that evolution here, but I'm going to talk about evolution fairly often during the course. And the reason I'm bringing evolution into a course on biochemistry is because it is the broadest, most explanatory tool we have for making sense of biochemistry. It really is a wonderful integration for biochemistry. And if there are any of you who are concerned about that, who are uncomfortable with thinking about evolution as part of all of this, I want to reassure you that there's no necessary conflict between evolution and whatever religious perspective you might want to bring to things. If you want to talk about that with me sometime, I'm quite open to discussing it. But I'm not going to back off. Things evolved. The oldest cells were around 3.5 billion years ago. And the kinds of things that we see are evidences that for both the eubacteria, including the cyanobacteria, and these archaea, quite far back into the order of 3.5 billion years, the eukaryotic cells have diversified into the kinds of things that you recognize by seeing, cell, animals, plants, and so forth. And there are certain features that we share as eukaryotes with the group that are called archaea, and I have more references for that if you want to see some of that. So there are some features where we resemble archaea more, a few others where we resemble bacteria more. It really does make a sort of a juicy story about exactly how did the eukaryotes emerge from the prokaryotes. And we have both biochemical clues to that, where the chemistries are similar and so on, and also structural ones to some case. Now, one proposal that's been made is that chloroplasts, this is from your text, you see, this is the kind of the hippie commune approach to the eukaryotic cell. And the idea here is that a blue-green alga and some kind of a bacterium all got together and made a cooperative cell by each of them becoming part of this commune, or whatever you want to call it. And it's true that the chloroplasts and the mitochondria have DNA, so that is certainly a clue to something about where they came from. But on the other hand, eukaryotic cells can make compartments of considerable complexity that don't have any genes. And another difficulty that I have with this theory is that it's trying to propose that an anaerobic cell ate the mitochondria and the chloroplasts. And an anaerobic cell doesn't have much energy because it's anaerobic. If you look among the eukaryotes, the cells that can eat other cells are aerobic. And when they're eating another cell, they're metabolizing at the fastest they ever metabolize. So phagocytizing something is a very expensive activity, and I have difficulties with the idea that an anaerobe would have the punch to stand up and eat that guy. There are alternative theories available. This is one that's been proposed. This is showing actually a proposal for the evolution of the nucleus. And the point that they're making here is that if we look at prokaryotic cells as we know them today, they have their DNA attached to the cell membrane at a structure called a mesosome. And they have membrane-bound ribosomes that are making proteins for the membrane and for the extracellular space. So the proposal is that by just geometric rearrangements, you could start to make a compartment around the DNA. You can imagine the cell membrane invaginating here and here to create this kind of bay for the DNA, and the ribosomes remain there on that membrane. And it's just a little bit more of a geometric rearrangement to get from there to there. And so this is a proposal for how a nucleus and an endoplasmic reticulum might have arisen. It, to me, is not very much of a leap to suggest that possibly there was another compartment like this, but it specialized in photosynthesis and had differential gene loss or something like that. So I think the most honest answer is we don't know exactly how the eukaryotic cell evolved. We have complete confidence that it has some relationship to the prokaryotes because there's much too much similarity. But we don't have a knockdown, drag them out argument for exactly how that happened. It probably happened around 1.5 billion years ago, but there was a big burst of oxygen around 500 million years ago, more or less, 600 million years ago of oxygen, and it's at around that time that you see in the fossil record not just eukaryotic cells, which we can find back 1.5 billion years, but multicells. Being a worm is energetically more expensive than being an amoeba. Sure.
So to do a lateral transfer,
I wouldn't say I find that the most persuasive. So in other words, you're talking about, sorry. Do we have a figure on that here? Is that the one you're talking about here? Oops.
Yeah, I mean, like, we talked about that in evolution, so I was just wondering.
Right. There are certainly difficulties with that theory. And I would say, if you're interested in knowing more of the details about things like that, I would recommend Karen Smith, who is a British scientist who has done very detailed examinations of this. I think that the lateral transfer theory, if you just take it at one level, is straightforward enough to be very attractive. But then if you look at more details, it starts to get more complicated. And Karen Smith is my preference, my source. But even he would admit that it's not, crystal clear which one is true. Is there an interest by putting Karen Smith's article up? Would people show their hands if they'd like it? Okay, that's enough to justify it. So I'll do that, and I'll have some other things as well. Now, I think we probably should leave the rest of these, what's rest that's left. What's left here are some of the figures from the study questions, so that these are a way to take a look and get a sense of the various groups. As I said, you've met all of these before. It's imperative for you to feel completely comfortable with them, because these are the vocabulary we'll be speaking in the language of biochemistry. So if you have any hesitation about looking at these things and knowing what they are, or hearing the word alcohol and knowing what it feels like, refresh them. These are some more of those groups, and the rest of them. Okay. So those are done up big enough that you can look at them and draw on them and so forth. I think you have the handout for Chapter 2, is that correct? Yes? Does anybody need the handout for Chapter 2? Okay. We'll just start it a little early. Let's see, I need to go back. All right. Oh, before we get distracted and so forth, this is a nice natural time to break up things. Angie Henson had a comment she wanted to make. Do you want to make it yourself, Angie?
Yeah.
Okay. Or I can do it from here. In the announcement that we sent around about the office hours, we mentioned that her Thursday evening sessions will start February 1st. But she took a look at how long that is in the calendar and thought she'd like to offer a session on this coming Monday at the same time?
I don't know.
Okay.
Probably around 5:30.
Okay. Okay. So she'll have the details for that session on Monday at the Monday lecture. I know that's not a close time to plan ahead, but she did want you to know that there's that opportunity there, so kind of mentally keep a space for it if you think you'd like to do that. Okay. So let's move on to water. So Chapter 2 is sort of our first part of thinking about structural biochemistry. And oddly enough, the first thing we think about for structural biochemistry is water, which is almost the simplest molecule you could think of. This little question and answer at the top was the opening overhead in my class of biochemistry of what would life be like without water? Crisp. Just think of potato chips. And there are a number of reasons why water is essential for life, and why any life anywhere in the universe would probably use water as its basis. First of all, it's an amazing solvent. Water as a solvent can dissolve probably the widest range of compounds that we know. And we'll see why that's true in a second. Secondly, its properties give rise to the basis for membranes and proteins to have structure, and we'll see what the basis for that is. Third of all, it's due to the structural properties of the water molecule that acid and base relationships hold for living systems. There are, of course, acids and bases in non-aqueous systems, but we're going to be thinking about it in this one because this is where living things are. And because water does all these things so well, life is tied to living water. It is tied to liquid water. That you find the temperature limits to living systems bracketed by where there's liquid water. So once it's solid, there's no metabolism going on. Once it's a gas, no life going on. And it also limits the chemistry that can happen. So there's some interesting things that are out there in the rocks and stuff. Living systems can't take advantage of it because they are based in a water-based system. I think it's interesting that for the astronomers who are trying to see life elsewhere, one of the strategies is to look for the spectral profile of water in the planets out there. We have a lot of water in us. About 60% of our mass is just water. And any given cell, it tends to be about 50% water. And what's interesting about that is that it's only 50%, that it's half something else. If you talk to an X-ray crystallographer, a protein crystal is also often about 50% water. And there are protein crystals that have even more water in it. So cells are permeated by water, but we're not awash in it. It's not a dilute solution. It's a very condensed solution. If we were to look at the makeup of the various components inside the cell, we have about 40% water. We have outside the cell interstitial fluids, about 15%. Another 5% in the circulatory system. Interstitial means between the cells, not actually in vessels, but just between the cells. And then about 40% carbon, hydrogen, nitrogen, proteins, carbohydrates, stuff like that. Now, the reason water is so amazing has to do with its electronic structure. And it looks very simple, but it's kind of like a Shakespeare sonnet. It repays close attention. So you have in the middle here your oxygen atom, and you have here a hydrogen and here a hydrogen. And the electron clouds coming off of the oxygen are in this tetrahedral arrangement. So you have the hydrogens coming off at an angle like that, and you have these other two clouds of electrons that are sticking out the backside. Now we talked about electronegativity, and if you remember, the hydrogen was the least electronegative and oxygen was the most electronegative. So although these are covalent bonds, they don't share equally. Oxygen is greedy. And so there's a partial negative charge on the oxygen and a partial positive charge on the hydrogen, because the oxygen is winning, it's on its way to winning the tug of war. It doesn't quite win it there. And so we talk about water as, I know what I have to do. I have to tell the pointer to, and so what that means is it's sort of like a Velcro situation. You've got a little bit of positive here and here, and you've got a little bit of negative back there. You can make bonds between those that are called hydrogen bonds, and it makes the water molecule as a whole polar. The bonds between the oxygen and the hydrogen are polar. The whole molecule is also polar because the hydrogens are to one side and not the other. So the bonds are polar and the molecule is polar. And that overall gives us a chance to make truly interesting structures when we look at liquid water. And this is just actually drawing in that those partial charges for you, the hydrogens and the carbons. And I've actually anticipated myself. These are polar bonds, and then overall you can see how all of the positive charges to one end and the negative charge is to the other. They are partial charges, but there they are. And this is showing then what the structure of a hydrogen bond might look like. Here you have your positively charged, partial positive. So the delta here, the Greek delta, means partial, a bit of positive charge, a bit of negative charge on the oxygen. And so opposites attract, and the hydrogen will tend to point towards that oxygen and spend more time there. It tends to hang out there. The better this alignment is, the stronger that relationship will be. And there are some examples where when we look at these hydrogen bonds, there isn't a short side and a long side as we see here, which is probably the more typical type of hydrogen bond. But they're so exactly shared that the hydrogen is in the middle, is equidistant between the two. And that's called a low barrier hydrogen bond, where that hydrogen could be on either place about equivalently easily. And that tends to be a bit shorter, and it tends to therefore be a bit stronger. This hydrogen bonding capacity is what gives rise to the structure of both water and ice. Water can be the donor on a hydrogen bond, it can be the receiver on a hydrogen bond, so absolutely pure water can be completely bonded to itself, not to the individual molecule, but one to another. And if we look at the structure of ice, for instance, we can see that every potential hydrogen bond is formed, that there are 4 hydrogen bonds per molecule. And if you look at that, you can see it's a rather open packing of it. The openness of this packing is in a bit of contrast to the liquid water, where it can kind of scrunch around a little and reach behind. In liquid water, it gets close to 4 per molecule. It's not quite there. The hydrogen bonds are much shorter lived than the water ones. And one of the consequences of the open packing of the ice compared to the water is that ice is almost unique, or perhaps unique, in having a solid that is less dense than the liquid. Which says, so what? My ice cubes float. Well, yeah, they do. Fortunately, the ice in the ocean floats. If ice didn't float, when ice formed, it would go to the bottom of the ocean and stay there. And we would fill up from the bottom and have nothing but ice. So the fact that ice floats is one of the things that contributes to the possibility of life on Earth. Now, although these hydrogen bonds in liquid water are not as long-lived as the ones in ice, they are enough to make water cohesive to itself. And we'll talk more on Monday about the implications of that. See you all Monday. Have a great weekend.
Thanks.
Biology.
cells have sugar across a lot of sugar across. What else actually, the membrane proteins themselves have sugar. And those sugars are different in different